So the last technique I want to talk about with respect to biotechnology relates to an operon that's found in E. coli called the Lac Operon. And it's a really good example of how our knowledge about the various processes of molecular biology allow us to develop very powerful tools to use in the laboratory. So this particular operon, we don't need to worry too much about the details, contains a gene called the Beta-Galactosidase gene. And the Beta-Galactosidase gene has a very useful property. In an E. coli cell, the normal function of the Beta-Galactosidase gene is to cut a bond in the sugar known as lactose. And that cutting of the bond of lactose breaks lactose down into galactose and glucose, the two sugars that comprise it. And that's not really important for this purpose, actually. Scientists have developed an artificial chemical that beta-galactosidase can work on. It's called X Gal. And X Gal, the enzyme treats as if it's lactose, and it cleaves a bond in X Gal. And the advantage of that is that when it cleaves that bond, it turns X Gal into a molecule that has a blue color. And so it's very easy to see which cells have beta-galactosidase by treating them with X Gal, because if you do that, then any cells that have beta-galactosidase that's being made will have a blue color because the beta-galactosidase will act on the X Gal to give that blue color. So what researchers have done with that is they've taken and they've developed strains of E. coli that contain no beta-galactosidase, so that if you treated those cells with the X Gal, they would not produce a blue color. In fact, they would appear to be white. On the other hand, if you introduce that beta-galactosidase gene into those cells and treat them with X Gal, they will turn blue. So, well, that's fairly easy to do. I've talked about recombinant DNA and how one can put a gene like X Gal into a plasmid and then put that plasmid into bacteria, and so bacteria make X Gal, and make beta-galactosidase, and you treat them with X Gal and they turn blue. Well, there's one additional step that people have used with X Gal that help them to determine whether or not they've made a recombinant that they want. And so that's what's the subject of the next slide. So in this slide, we can see on the left a plasmid like what I've described. That is, this plasmid has contained within it the X Gal gene. So if I take these E. coli cells that have no X Gal, and I put this plasmid that's on the left over here into the cells, then what will happen is they'll turn blue when I treat them with X Gal. Well, that's not the important point of this slide. So let's imagine that I have a recombinant DNA that I want to put into a plasmid. And the plasmid that I have here is this artificially made plasmid that contains the beta-galactosidase gene. It turns out that there's a complication with respect to this that I haven't really described. So this figure begins to illustrate that. There's a couple of things that we don't want when we try to make a recombinant DNA. And those things that we don't want, thanks to this technology I've just described to you, help us to tell which results are good and which results are the ones we don't want. So let's say I've got this gene, let's say it's a human growth hormone, and I want to insert it into a plasmid so that I can make copies of it and so forth. So I've got my gene, it's on a piece of DNA, it's been cut with a restriction enzyme. And I have my plasmid that's over here that has the beta-galactosidase gene in it. I cut the beta-galactosidase gene with the same restriction enzyme, which happens to cut in the middle of that plasmid, okay? So what's going to happen when I cut it? The beta-galactosidase gene will be cut right in its middle, and it'll be opened up so that now instead of being a circular DNA, it's going to be a linear DNA, and the ends are each going to have the beta-galactosidase gene there. If I take my gene that I want to insert into there, I mix it with this linearized plasmid, and I add DNA ligase, what's going to happen? Well, DNA ligase, of course, catalyzes the joining of ends. And so a couple possible things will happen. Number one thing that can happen is that the DNA ligase is simply going to take the ends of the plasmid and put them back together, meaning no insert. Well, that's not what I want because, of course, I want to have an insert. That's the result I'll get from the very top slide that you can see here. On the other hand, if I put the, if the DNA ligase takes that gene that I want to insert and ligates it to the ends of the beta-galactosidase that I had linearized, I'll make a circle now that contains that human growth hormone gene that I want to insert. So two possibilities. I've got a plasmid that contains no insert. I've got a plasmid that contains an insert. Now if I take that mixture, because I can't tell which one's which, I take this mixture after I've ligated it and I add it to E. coli cells that don't contain beta-galactosidase, what's going to happen? Well, it turns out that there's a third thing that can happen that happens in any cloning result. So that third thing that can happen is that some cells are going to take up plasmids. Some of those cells are going to get the beta-galactosidase gene intact, that is no insert. Some cells are going to get the DNA that contains the insert down here, and some cells are not going to get DNA at all. In fact, that's the most common result that you get. Well, one of the tricks that we use in introducing DNA into cells with plasmids is that the plasmid contains resistance to an antibiotic marker, like commonly ampicillin, for example, so that any cells that get the plasmid will grow in medium that contains ampicillin, okay? So the three possibilities then that I can get for any experiment that I've just described to you are shown as follows. So let's start, for example, with cells that don't get the plasmid. When I introduce this mixture, the vast majority of cells will not get the plasmid. I take this mixture of cells that I've introduced the DNA to and I smear them across a plate that contains ampicillin. In the case of the middle situation where cells don't get the plasmid, there will be no growth because they are sensitive to ampicillin. They will die in the presence of ampicillin and we will never see those cells. The second possibility is that we will get cells that get the plasma but contain no insert. Well, they're going to be resistant to ampicillin, so we will get some colonies that will grow. And if we treat those colonies with X Gal, what are we going to see? Well, in this case, the beta-galactosidase gene came back together intact. It's functional. And so these cells that are here are going to be blue because beta-galactosidase will cleave the X Gal and produce a blue color. Now, I don't want these cells because, of course, it has no insert. And these cells I don't want, and of course those cells are all dead, so I don't even need to worry about them. But the cells that I want are down here. They're the ones that contain the insert. Well, what happened when I put that insert in the middle of the beta-galactosidase gene? Well, in fact, what happens when you do that is you disrupt and destroy the function of that beta-galactosidase gene. Consequently, if I treat these bottom cells with X Gal, what I will see is not blue color because there's no beta-galactosidase that's functional. Instead, I will see white color. So the beauty of this assay is I can tell at a very quick glance upon treating cells with X Gal by simply looking at the color which cells contain a plasmid that contain an insert. And it will be these down here that are white.